In this video, we will finalize our discussion of the second line of defense by looking at some non-specific chemical defenses, including the interferons, complement, inflammation, and fever. In addition to the defensive cells, the second line of defense employs several chemicals to destroy pathogens. To defend itself against viral pathogens, human cells produce small molecular weight chemicals known as interferon. Interferons act to interfere with the viral replication cycle. Interferons are not virus-specific, meaning that they are effective against a number of different viruses. Different interferons are produced by different cell types. Type 1 interferons include both alpha and beta interferon and are produced early in an infection, while type 2 or gamma interferon is produced later in an infection. Type 1 are produced by virus-infected cells in very small quantities and diffuse to uninfected neighboring cells. The interferon binds with receptors on the surface of neighboring cells to elicit a series of reactions that result in transcription of genes in the nucleus. The mRNAs produced are then translated to produce antiviral proteins. Antiviral proteins act to disrupt various steps in the viral replication cycle. Type 2 interferons are produced by T lymphocytes and natural killer lymphocytes. The secretion of type 2 interferons from these cells acts to stimulate neutrophils and macrophages to kill bacteria. Please be aware that interferons cause malaise, muscle aches, chills, headache, and fever, which are all typically associated with viral infections. During our discussion of the defensive components of the blood, we briefly mentioned the plasma proteins of the complement system. Here we will examine them in more detail. The complement system, which is usually just referred to as complement, is a defensive system of over 30 different plasma proteins. Its name is derived from the fact that it complements the activities of the defensive cells of the immune system. The complement system may be activated by three separate pathways: the classical pathway, the alternate pathway, and the lectin pathway. The classical pathway is named because it was the first one to be described. It is activated when antibodies bind to microbes. Antibodies bind to antigens on the surface of the microbe to form an antibody-antigen complex, which is then bound by complement protein C1. Activated C1 activates C2 and C4 by splitting them. C2a and C4b combined and together activate C3 by splitting it into C3a and C3b. C3b binds to the surface of microbial cells and serves as an opsonin to enhance phagocytosis. C3b also activates C5 by splitting it into C5a and C5b. C5b then binds with C6, C7, and C8 and then inserts themselves on the microbial cell membrane, where they then attract multiple C9 proteins that form a pore in the cell membrane, known as the membrane attack complex. The image on the right shows a single bacterial cell with numerous membrane attack complexes, seen by the small black circles. The many membrane attack complexes result in the lysis of the cell. The alternative pathway is activated by the presence of pathogens or pathogenic products, rather than the presence of antibodies. The C3 protein is constantly present in the blood, as well as some other proteins known as factor B, factor D, and factor propyridine. Factors B, D, and P are attracted to molecules on the surface of pathogens. When they bind to the pathogen and interact, it causes C3 to be split into C3a and C3b. The results will then be the same as seen in the classical pathway. The lectin pathway is activated by the presence of mannose sugars found on the surfaces of bacteria, fungi, and some viruses. Mannose is rarely found on mammalian cells. Lectins are proteins produced by the liver that can bind to carbohydrates. Mannose-binding lectins bind to mannose molecules in order to activate complement. When mannose-binding lectin is bound to mannose, it cleaves C2 and C4. The cascade then continues to produce the same results as seen in the classical pathway. Inflammation is the body's nonspecific response to tissue damage. Inflammation is generally characterized by 4 signs and symptoms: redness, heat, swelling, and pain. Inflammation may be acute or chronic. Acute inflammation develops quickly and is short-lived. Chronic inflammation is long-lasting and causes damage or death to host tissues. We will focus our attention to the events of acute inflammation. Acute inflammation is an immediate, nonspecific response to damage to the body's tissues. Inflammation may develop following physical injury or chemical injury, or may result following infection. Inflammation serves several functions. First, to destroy or remove the agent that has caused the damage, and secondly, to repair the damage that was caused to the tissues. Your textbook addresses 3 aspects of inflammation: dilation and increased permeability of blood vessels, migration of phagocytes, and tissue repair. Following invasion of pathogens, blood vessels within the affected region dilate, thereby increasing blood flow to the area. This characteristic of inflammation is responsible for the redness and heat associated with inflammation. The permeability of the vessels within the affected region also increases, meaning that the cells lining blood vessels separate, providing more space for white blood cells to move from the blood circulation into surrounding tissue spaces. Vasodilation and increased permeability of the blood vessels is caused by several chemical substances, including histamines, kinins, prostaglandins, and leukotrienes. Increased blood flow due to vasodilation allows more leukocytes to arrive at the site. As the leukocytes arrive, they begin to stick to the endothelial lining of the blood vessel, a process known as margination. They then squeeze between the cells lining the blood vessel to move into the surrounding tissues, a process known as diapedesis. Once in the tissue spaces, the phagocytes can then begin to destroy bacteria. The final stage of inflammation is tissue repair. Tissue repairs begin to take place rapidly but cannot be completed until all harmful substances are removed or neutralized in the affected region. The ability to regenerate and repair the tissues is dependent on the type of tissue involved. Some tissues are repaired rather quickly due to the presence of stem cells. Such tissues usually can be returned to their original state. Other tissues that do not undergo regular cell division are unable to be repaired. Such tissues include cardiac muscle and brain tissue. The term fever refers to an abnormally high body temperature. The normal human body temperature is 37 degrees Celsius. Fever is stimulated by chemicals known as pyrogens. Pyrogens act on the hypothalamus of the brain, which is responsible for controlling the body's temperature. The hypothalamus basically acts as the thermostat of the body. Pyrogens reset the body's thermostat to a higher temperature. The body responds to this activity by constricting blood vessels of the skin, increasing the rate of metabolism, and stimulating repetitive muscle contractions that we commonly refer to as shivering. These activities together act to raise the body's temperature. Due to the decreased blood flow to the skin, the body feels cool to the touch, while the core of the body is heating up. This is referred to as the chill with a fever. Chills are a sign that the body temperature is rising. Pyrogens that act to raise the body temperature may be exogenous or endogenous. Exogenous pyrogens are those associated with pathogens and may be components of the pathogen itself or toxins produced by the pathogen. Endogenous pyrogens are chemicals released by leukocytes to stimulate fever. Fever is considered beneficial up to a certain point. Fever acts to inhibit the growth and activity of pathogens while at the same time enhancing the activity of phagocytes. If fevers become too high, they can cause damage to critical proteins that could lead to convulsions, coma, or even death.